This is David Gibson. I'm the executive editor for the Journal of Ecology, and I'm joined today by Bernhard Schmid from the University of Zurich. And we are recognizing Bernhard in the Journal of Ecology as our 2020 Eminent Ecologist. So this interview is an opportunity to learn a bit more about Bernhard and his background. So welcome, Bernhard.
Welcome, David.
So let's go back, sort of give everyone a bit of an idea of where you sort of come from academically, and tell us how you kind of got into ecology as a career.
Okay, that was at the Teachers Training College in Switzerland, where at the time we had lots of biology practicals. And in the last year of that education, I mapped all the herbaceous species in a 5-hectare wetland. And then I analyzed the data with the so-called phytosociology methods. So I got interested in plant ecology for that, and then studied botany in Zurich, working on a particular species, which gave me the most difficulties for mapping during this work at the training college. And this is Carex flava, yellow sedge, which has a lot of subspecies that actually differ in ecology and karyology. And later I found out Carex is really a very special genus because it has this kind of holocentric chromosomes that allows breakage and fusion without too much disturbance of meiosis, and therefore new species can very easily evolve. And I hope that people know that Carex is by far the most species-rich angiospermous genus in the world. So it's more than 2,500 species.
So you got interested in ecology by taking on a very difficult group of plants.
Yes, that's right.
Yeah. So at that time, were there people that inspired you?
Yes. So actually, when I had done this work in the wetlands, I asked my professor, it's Christopher Cook, who came from England to Zurich, what I could do, maybe in the way of continuing vegetation science. But his best advice was not to continue with plant sociology. And so he gave me all the freedom to study this Carex flava, which was my thing, and I could do everything on my own. He just protected me from doing any work for him. I could do whatever I wanted. And therefore, I somehow reinvented lots of plant demography and population biology because we didn't have any classes about this. But it was very good to be totally independent and develop things on my own.
Yes, that's very interesting. And so then when we met was when you came to Bangor to work in John Harper's lab as a postdoc, and I had just arrived, and I don't know whether you arrived first. I arrived as a PhD student to work with Peter Greg Smith in community ecology. So
that's right, yeah.
Our sort of interest started a little bit differently. But how did you come then to arrive in Bangor to work with John Harper?
Again, it was kind of not really the science. You know, when I had finished the PhD, I asked Chris Cook if he would have an idea for a postdoc in a remote place, because we had a small young family and we didn't want to go to a city. So he looked at the map and he came up with this place in North Wales, Bangor, where there was John Harper. And so when we arrived, you know, John told us after 6 months, you would either love or hate it. And fortunately, we really loved it. And I think still now North Wales was a beautiful place, has a wonderful landscape. It's spring from February to October. And in addition, there were really lots of people coming to Bangor for shorter or longer visits. So it was not necessary to go to a big place. And yeah, then there were some nice people there, like this PhD student from another professor who didn't mind to discuss research with a foreign postdoc who had just learned English by reading John Harper's textbook. Indeed, that was the only way I learned English.
My gosh.
And So this PhD is now you as a professor in the United States, presently interviewing me.
Well, yes, yeah, that was a long time ago. But yes, Bangor was a very special place for many of us, I think.
And so,
yeah.
I mean, I should of course say it was not only a coincidence that with John Harper, it was also that once I was there, he really got me interested in an academic career. So it was because of him that then after the postdoc, I was not thinking anymore of just returning to Switzerland and doing some job, but he inspired me, and also the intellectual environment in Bangor to continue. And afterwards I went to Harvard to work with Fachri Bassas on a further postdoc, and eventually got assistant professorship from Swiss National Science Foundation at Basel. And the interview for that was terrible. And later on, I found out that I probably only got it because Harper and Bassas, they wrote really very nice letters. So these things you usually don't find out, but I think we should be always very thankful to our advisors.
Absolutely, yes. And to me, Bangor was interesting because we had John Harper, population biology, and then we had Greg Smith, who was my advisor, community ecology, and they were sort of poles apart in terms of their ideas and how to do ecology and so forth. And yet most of their students and postdocs were somewhat in between, I think. And I got a lot from Harper and Greg Smith, and I think all of us probably did.
It was the same for me. I did learn a lot from Greg Smith. And, you know, even now when people do statistical, spatial ecology and so on, I think they often forget what Greg Smith and his people had already invented at that time. So I'm still quite impressed what they did in quantitative ecology.
Yeah. Well, we've already talked a bit about Carex flavor and your work on clonal plants, and so I wouldn't say you moved on, but you started then work on some other clonal plants like the solidagos, goldenrods, and things like that.
Yeah.
And so how does that arise? Do you switch plants, or do other groups of plants allow you to answer different questions?
Yeah, so it is a bit like this. When I studied in Zurich, it was more kind of what we called biosystematics, which was systematic botany, but you looked also at ecology, not only at morphology and karyology and so on. And at the end, when I finished my thesis about Carex flava, I realized that everything that I found could be seen in the light of R and K selection. So I actually gave this title to the thesis. But what puzzled me very much was that this life history strategies could either apply to the tillers of the Carex tussocs, or it could be the entire tussock, what we call the genet. And in Carex flava, it was kind of easy because both the life history of the tillers and of the tussocks showed R selection. But there are other Carex species that have, for example, very short-lived tillers and very long-lived genets. So, and this was, of course, at the same time, the lab of John Harper got very interested in clonal plants, and that's why somehow it was a fortunate coincidence. But in a way, we then realized that clonal plants just represent an extreme form of model organization, which is typical for all plants. So in a way, clonal plants are the best model to study plants. If you would bury a tree in sand so that only the twigs would stick out, it would just look like a clonal plant. And modularity is really what distinguishes plants from most higher animals. And that has very far-reaching consequences for ecology and for evolutionary theory. And I think many scientists now more than then prefer to work with annual plants because they are closest to unitary animals, and thus they allow you to apply theory developed for animals And then you can suggest that things are really general and encompass both plants and animals. But the clonality actually teaches us that plants are really different, because plants individuals as populations of their parts should rather be compared with animal populations than with animal individuals. And in a way, a plant is group living in perfection, because the leaves, shoots, and branches are genetic kin and physiologically integrated, yet at the same time they have a high independence and can react differentially to the local environment.
Yeah, I remember Harper talking about a tree being a population of buds.
Which is very true. Yeah, and the population of twigs, but even the population of leaves, you know, because leaves are basically behaving like individuals independently with a little bit of correlation with other leaves.
Yeah.
But if you measure photosynthesis, you stick a leaf into a cuvette, and it's more or less okay that you don't stick the whole tree into the cuvette.
Yeah. Okay, yeah, interesting. But of course, you haven't stuck with that sort of subject your whole career. You've, I don't know, maybe in the 2000s, maybe you can let me know here. You started to work on biodiversity ecosystem function questions.
Yeah.
So it's not a switch, but it's an addition to your work. Is that correct?
Yeah, in a way you could say it's a bit jumping on the bandwagon, but on the other hand, a lot of things in my career happened randomly. So I kind of stumbled into ecology, but then later I realized that the main questions were always, what is a community? That was at the very beginning, and what is the role of variability in nature? And I admit at first I was very fascinated by this plant sociology, because it was based on this Clementsian view of the community as a superorganism. And it's interesting that then, of course, people told me in my postdoc, oh, that's stupid. It's more like Gleason thought. Communities are associations of species that occur in the same place. And then I focused more on demography and population biology of clonal plants. I mean, I was interested in how the modular parts of an individual can work as an organism. It's also kind of a superorganism of parts. And when all these parts may actually function independently. So this led to this question, could diversity be a group strategy? And diversity could best be expressed as a group strategy if the parts are genetically related, as in a plant. So then, but when I came back to Switzerland, I had the chance to be involved in a so-called integrated biodiversity project. And there we focused on this possibility of diversity effects, not only actually at the ecosystem level, but even we went from the evolution of mutation rates in bacteria to relation between genetic variation and extinction or invasion probability in plants, and then finally to this biodiversity ecosystem function relation. And that's to somehow that then dominated my career since early 1990s. So we found these very strong positive effects of plant species richness on community productivity. And this was quite opposite to what we had learned from John Harper, who taught us that, and that was the sentence, under optimal conditions, the best monoculture should always be better than the best mixture. And actually, I made a handwritten note in a paper from Darwin and Wallace, 1858, where they said it has been shown mixtures are better, and I put several question marks in the margin. And now, of course, I deleted the question marks again, because now I see it's really true. And the good thing is that in the meantime, we found that it was this division of labor, which allows a community with several species to perform better than a community with a single species, because a single species cannot have this huge plasticity to do all things well. And what was the most exciting finding for me in the last 10 years was that when we analyzed the plants from the biodiversity experiment in Jena in the Gasland, we found they had within 8 years evolved increased character displacement and division of labor in mixtures. So probably because at the beginning the seed material was already containing variation, and this was kind of selected out, and therefore after 8 years you had already selected for this increased division of labor.
Mm-hmm. Yeah, okay, fascinating. So this brings us to your most recent paper in Journal of Ecology. You had several, and they'll be collected in the virtual issue that accompanies this interview. So this is a paper by Franca Bongas and colleagues, including you. And this, listening to your last answer, it seems that this is a continuation of these ideas, because here you're looking at intra-specific variability. The title of the paper, I'll just read this out so everyone knows, is Growth Trait Relationships in Subtropical Forests Are Stronger at Higher Diversity. Tell us a little bit about that study.
Yeah, so the origin of this project was kind of, again, like all the other things, coincidental. But the question was that we wanted to know, do these things that hold for meadows and for grassland also hold in forests? Because, for example, Steve Hubble with neutral theory said in these highly diverse tropical or subtropical forests, all species have the same niche, and there is nothing like this complementarity and division of labor. And therefore, we wanted to do the same for a tree experiment. We did this tree diversity experiment, started in China, and actually we found exactly the same effect of about 1.8-fold increase from monoculture to 16 species mixture in productivity. So it was really totally amazing. But what we also found, this was in this paper, is that in fact this individual variability and community diversity were positively correlated. So that is what we also found in the grassland experiment. But in the grassland experiment, it was due to this evolution of division of labor, whereas in the forest, it was more the plasticity that allowed the species in the mixture to separate out more. So, which is, I think, an interesting finding. But as I said, this collaboration with the Chinese colleague was kind of accidental, because we had a journey to Chinese wetlands. A German professor wanted to do some research there. And I was sent to accompany them, that they wouldn't do a stupid project. And so I told them, okay, let's suggest we do a forest biodiversity experiment. And then surprisingly, the German Science Foundation supported that, and also the Chinese Academy of Sciences. And so that's how now, since 15 years, I all the time travel to China, maybe twice a year, except now it was not possible. And we work on this big experiment.
Fascinating. Yes, well, I'm glad you published that paper in Journal of Ecology. So as we bring this to a conclusion, you've had several papers in Journal of Ecology. Are there any other ones that you want to just mention briefly?
I hesitate between the one by Lisa Marquardt, where we showed that biodiversity in grassland increased productivity mainly because mixtures allow higher total densities than monocultures. And the other one, which was demographic analysis of a single population of goldenrod, Solidago altissima. And this, in my view, was the most comprehensive demography study in plants after Jose Sauerbrann's study of this ranunculus species in a pasture in North Wales. And Andrea Meyer, who did this work, actually, for example, excavated rhizomes every year and buried them again to see how these plants grew underground. And in the end, he produced these perfect figures according to the book, The Visual Display of Quantitative Information by Edward Tufte. So I'm sure these are the graphs in Journal of Ecology with the highest data to non-data ink ratio.
All right. Well,
you can check it out.
Yeah, we will. Yeah. Okay. Oh, good. Yeah, fascinating. Okay. So finally, then, what advice would you give as a senior ecologist to early career ecologists that are listening to you today?
Yeah, okay, so of course now it's different. At the beginning of my own career, ecological research was typically done individually or in small groups. And I think I was really lucky to have been able to do my PhD at that time. In contrast, my own master and PhD students were included usually in big projects, which became ever more complex. And thus, they had to work on a particular, more specialized topic within a large team, which probably will be the common situation in ecology also in the future. So under these circumstances, my advice is to engage actively in collaboration and exchange data freely with others, as freely as possible. So because if you keep a variable x1 and the other one variable x2 for yourself, then you miss the opportunity to combine the two variables in a synthesis, for example, looking at correlations and trade-offs. And another more prosaic advice is to learn statistics from basic principles and mistrust everything that you cannot understand yourself. I see a great danger that ecologists use statistical tools inappropriately because they follow these general recommendations rather than their intuition and logic. My impression, for example, is that mixed models, and I would like to write a paper about this, are wrongly specified in the large majority of all peer-reviewed ecological papers. But I admit that it takes a lot of learning and experience to avoid such pitfalls. But if you do learn that, you will be richly rewarded.
Okay, so very good advice for everybody. Yeah, well, thank you very much, and thank you very much for this chat. I've enjoyed it, and I know that people listening will enjoy hearing what you've had to say today. So thank you very much.
Thank you too. It was very nice.